Any student who has the drive, who has the passion, we want to give them the opportunity to succeed. So get their foot in the door, that first step, which is a job, but we are also looking at really getting them ready to succeed in the career. And we are doing this all through the internship part. So we're really rethinking internship as a way to expand opportunities and make the STEM force more inclusive. And I'm an optimist, a diehard optimist. I believe in the STEM community. I deeply believe in students. So looking at the summer numbers, it's no longer just optimism. We have numbers which prove that we are going towards these goals. We can make STEM more inclusive and really give opportunities to students. And I'm also an engineer. So I was at Intel doing something totally different. The reason I left Intel to start STEM Away was I had this idea about how we can achieve it. And that's where the mentor change comes in.
The Bioinformatics Pathway was driven by hands-on learning. Leads and mentors gave participants the necessary tools and access to information to learn independently and at their own pace, while still abiding by the internal team set goals. We did so by stressing communication, organizational, and troubleshooting skills to empower participants to take their own initiative to learn, creatively problem solve, and complete the tasks at hand. The final result was end-to-end execution involving both lateral and strategic thinking. Through the Bioinformatics Pathway, we wanted our participants to first learn the skills and then the application. So what that means is that we wanted to give them the necessary tools and skills to learn the overall pipeline and then be able to apply it to any scientific paper in order to reproduce the results. We had 3 main steps in the overall pipeline, as you can see here: normalization and quality control, DGE analysis, and finally functional analysis. More details will be covered in the following slides, as each lead will talk about a specific section of the pipeline, which will set the stage nicely for our panel discussion at the end of the presentation. Also, during the discussions, we welcome anyone to join in with questions or comments. Once participants had a fundamental understanding of the overall pipeline, we asked teams to apply it to the following research paper and reproduce selected results. Just to give a brief background on the paper, this study analyzed microarray data to explore potential prognostic markers in colorectal cancer via the construction and in-depth analysis of a competing endogenous RNA network.
The next step in quality control and data processing was to normalize our data and perform background correction. A background adjustment must be performed to remove signal due to non-specific hybridization, that is, signal emitted by other things than sample hybridized to probe, while normalization of data must be done to correct systematic biases due to causes such as different dye assumption, spatial heterogeneity in the chip, or other stuff. As we know, performing a microarray experiment is pretty difficult, so we can have this kind of errors. Then, what do you see here? What you see here are just 2 PCA plots. On the left, you see the PCA plot of the unnormalized data, and on the right, you see the PCA plot of the normalized data. We do this to show the participants that normalizing data is pretty important, because from the first plot, we could say that sample 11 is an outlier just because it is outside from the ellipse, including all the samples in this cluster. And we will also say sample 21 and 26 are outliers. But after normalization, what we realize is that sample 11 and 34 are the real outliers here.
As Yves just mentioned, we use project management tools to work on the project as efficiently as possible. Our main method of communication with the interns was through the STEM Away forums. G Suite was also very important for us. We used Google Calendar extensively to schedule our meetings. All the leads would meet on Sunday afternoon and plan out the meetings for the week. This ensured that everything was posted and ready to go at the start of the week. Deliverables, meeting recordings, and notes were posted on Google Drive. And we used Slack for emergency communications in case the meeting link was broken or for quick reminders to turn in deliverables. We used Drive as leads to do the weekly surveys Aaron had mentioned earlier. These surveys help gather feedback on the intern's experience. The overwhelming majority of the feedback on these surveys was positive. However, there were a few negative responses as well. This was quite a shock for us since it was our first time dealing with negative feedback. we were putting in a lot of work and weren't sure what else we could do as leads. So we had a talk with Ablita, who made us feel better and showed us to focus more on the positive and take negative feedback as constructive criticism. We also introduced GitHub because we needed a way for students to submit their codes. However, it proved to be an additional complexity that some students were struggling with, so we made it optional. Students could still submit code via the Google Drive as they had done before, but at the same time, they got introduced to the basics of GitHub, which is an essential tool for future data scientists.
The next step on the pipeline was the differential gene expression analysis. And so the differentially expressed genes were ones that had significant difference in read counts between the different conditions, in this case, cancer or non-cancer. And so the main question we had was, what was the best way to standardize this analysis and which tools could we use? So we talked extensively to the mentors who had kind of come up with a good pipeline for us, and they had told us to use the LIMA package. And so that proved to be a really great way to do the DGE analysis. One of the biggest challenges here, though, was that this was kind of a really big step up from the previous packages they had used because it was so extensive. And so one of the ways that we kind of combated that was by having more frequent office hours and actually inviting the mentors to show up to those as well, so they could give more of a deep insight into the packages and what their use was. And so here you can kind of see the three main steps we took for that week of the DGE analysis. First was the annotation step, which was conducted on the data to match the probe IDs to the gene symbols. Next was gene filtration, which was used to remove non-relevant genes. And then finally was the LIMA analysis, which was the R package we used to generate a design matrix and then form a linear model. So now this is just another slide showing why we use LIMA. Basically, it allowed us to account for variations in the data quality, accommodate technical and biological heterogeneity, and reduce noise, like Yves was mentioning earlier.
Once we have identified the top dysregulated genes through differential analysis, we can give them a more applicable biological meaning through functional analysis. In this step, we identify the most common cellular processes and functions related to our dysregulated genes and identify the roles of proteins these genes code for. Throughout the course of the pipeline, we use many different tools for functional analysis. The first of these is gene ontology analysis, using the enrich.go function in R. Gene ontology uses a comprehensive model of biological systems to identify the top biological processes, cellular components, and molecular functions associated with dysregulated genes. It is often useful to compare differences between overexpressed and underexpressed genes because it is the imbalance between the roles of up and down regulated genes that leads to the progression of disease. For example, in the case of colorectal cancer, which we studied during the internship and is shown in the bar graphs, we see that many upregulated genes are involved in RNA metabolism and catabolism, while many downregulated genes are involved in signaling pathways. This makes sense because in the cell cycle, RNA primers are built and broken down during DNA replication and intercellular signaling decreases. And these different processes help explain the continual replication of tumor cells.
Following the gene session, we wanted to bring more of the biological aspects of bioinformatics to the forefront. And the first tool we used was the string database. The string database contains known and predicted protein-protein interactions, or PPIs, including direct and indirect associations. Given a list of differentially expressed genes, the web application for this database constructs a network of genes and their PPIs with other genes. The primary purpose of using the string database was to identify targetable genes based on their interactions. We can identify these genes by comparing the number of connections each gene has. On the plot, those with a greater number of connections are key drivers. So for example, on this network, we have 2 clusters of genes with an average connection count of 6, and then one gene in the middle called MYC, or MYC, with 20 connections. So in this case, MYC would be considered a key driver. MYC is a proto-oncogene that codes a protein involved in cell cycle progression, apoptosis, and cellular transformation, which are all processes that are known to be abnormal in cancer. Following the identification of key drivers, we looked at global gene set enrichment analysis, or GC. This analysis, unlike gene ontology, looks at enrichment across all the genes in the pathway. It doesn't require cutoff values, and so is considered unsupervised, and allows us to investigate the additive effects at a systems biology level.